It's my pleasure to introduce the FOX3 Discovery Platform and Molecular Guidance System technology developed at SRI International. The FOX3 moniker comes from the NATO code of the same name, which indicates the release of a radar-guided missile from air to ground. By analogy, we are developing molecular guidance systems that allow for systemic delivery of a therapeutic that can then home in on a particular cell type or tissue with precision. FOX3 is a high-throughput selection platform that identifies peptides, or as we refer to them, molecular guidance systems, or MGSs for short, that have three key features. The first is that they bind to their desired cell type with high affinity and high cell specificity, much like monoclonal antibodies. However, upon binding, these MGSs trigger rapid internalization, allowing us to deliver cargo intracellularly. Furthermore, we've worked out selection pressures that allow us to identify MGSs that traffic to particular subcellular locations. This allows us to deliver a therapeutic to the correct subcellular location and gives us a level of precision delivery that is not available with other targeting modalities. While the MGSs can be used much like antibody drug conjugates in opening the therapeutic indices of small molecules, we think the real sweet spot for this technology is for the delivery of biotherapeutics such as peptides, proteins, antibodies, and nucleic acids that cannot reach the internal workings of the cell without a delivery vehicle. This allows us to now start thinking of these therapeutic modalities for new intracellular targets that were previously inaccessible. The FOX3 molecular guidance system has several key advantages. First, the platform itself is very rapid, allowing us to identify MGSs within 4 to 6 weeks from a full library to a lead candidate in hand. Because the cellular uptake and trafficking are incorporated into the selection process, no time is wasted on non-internalizing ligands. The MGSs identified are highly specific, able to distinguish between very closely related cell types. And the precision delivery to a subcellular location allows us to improve the activity of biotherapeutics. It is highly effective with internalization half-lives in the order of minutes and allows us to reach intracellular concentrations up to the low micromolar range. Furthermore, we've shown that the MGSs have the affinity, specificity, and stability necessary for in vivo delivery. Finally, the process is extremely versatile, adaptable to numerous payloads as well as numerous different cell types. Through the years, we've identified numerous MGSs to panels of different cell types. The majority of our efforts have been focused in oncology, where we have developed 25 different MGSs for solid tumors. Of these 25 MGSs, many have been optimized and validated, and we have MGSs that target 5 different subcellular locations and have shown delivery of 8 different payload types. However, the process can be used on virtually any cell, and we have many examples of targeting other cell types as shown here on the slide, including cells of the immune system, beta cells of the islets of Langerhans, and cardiomyocytes for cardiovascular disease. Additionally, we have validated the delivery of multiple different types of payload. The MGSs are able to deliver a wide variety of cargoes, including small molecules up to large full-length IgGs and nanoparticles greater than 100 nanometers. Because these are peptide-based targeting agents, we have the full arsenal of synthetic chemistry at our hands in order to conjugate different cargoes easily. Furthermore, we know a lot about how to modify these MGSs while maintaining their cell binding and internalization. SRI is using these MGSs for numerous applications, and we have several current programs ongoing. I'd like to highlight just 2 here, the first being the delivery of modulators of protein-protein interactions, and in particular monoclonal antibodies. We have examples of delivery of full-length IgG inside the cell that can then adapt to and modulate cell signaling. For example, we have delivered a function-blocking KRAS antibody that is able to block KRAS signaling in vitro and blunt tumor growth in vivo. Next, I'd like to highlight our efforts in delivering nucleic acids, with early programs showing that we can deliver siRNAs in a cell-specific fashion. This technology is limited really only by the types of cells you would like to target and the payloads delivered. We are continuing to expand the approach by developing MGSs for new cell types as well as novel cargoes. Thank you.